Miss, are you saying something?
Yeah, if you guys can't hear me yet, can you guys hear me now?
Yeah, it works now.
All right. Okay, so right. I was saying that today we're going to be starting off with the topic of enzymes, right? Enzymes are basically biological catalysts that are, of course, going to be speeding up those reactions that are taking place within our body. Now, when we talk about enzymes, we can classify enzymes in multiple ways. One of the ways in which we can classify enzymes is we can have intracellular enzymes and we can have extracellular. Intracellular enzymes are those enzymes that are going to be functioning within the cell, meaning that they're going to be working within the cell itself. So, for example, they're going to be responsible for breaking down your bacteria, right? They're going to be responsible for breaking down your food particles. They're going to be responsible for digesting the stuff inside the cell, destroying old organs that are not functioning anymore. If you want to destroy them, you would need cells that are working inside. So that would include your digestive enzymes, hydrolytic enzymes, basically, mostly. Then another type of enzyme that you are going to find yourself dealing with is, of course, going to be extracellular enzymes. Extracellular enzymes are those enzymes that are those enzymes that are actually going to be working outside the cell, meaning that they have to be released from the cell. So a good example of this that you guys might remember from your previous topic, from your IGCSE syllabus, is that a really good topic that we used to have was that we used to have these digestive enzymes that's released by the pancreas. You guys remember, in the topic of digestion, you might have studied it, that the pancreas release your digestive enzymes, your peptidase, your maltase, your trypsin. All of these enzymes were released, your lipases, they were released by what? The pancreas. So the cells of pancreas were producing these enzymes, and these enzymes were then released from the pancreas, and they were then carried on from the pancreatic duct into the small. That is an example of extracellular enzyme because that enzyme has been released outside the cell. It's traveling through a duct and it's going to the intestine, right? So those cells that are going to be functioning outside the cell, right? Those enzymes are going to be known as extracellular enzymes, and those enzymes that are going to be functioning within the cells are your intracellular enzymes. So intracellular enzymes will include hydrolytic enzymes that are going to be breaking down bacteria, food particles, old organelles, all of that. While extracellular enzymes will include those particular enzymes that are going to be responsible for that are going to be responsible for digesting those substances that are outside the cell. So a big example of this is your digestive enzyme, right? Your peptidase, your amylase, your maltase that are being released by the pancreas. That's a big example of it. So those are extracellular enzymes. Now, when we talk about how the enzymes are going to be functioning, so there is one theory that you guys have probably studied before, and that's the lock and key theory. The lock and key theory is an older theory. We now have a better theory, a more newer theory that we follow, and that's known as the induced trick model. Let's discuss what these two theories are, what these models are. So, first of all, what are enzymes? Enzymes are proteins, right? Enzymes are proteins, and as you know, enzymes generally have up to a tertiary structure. And because of the fact that the hydrophobic R groups are going to be facing inwards, the hydrophilic R groups are going to be facing outwards, the enzymes are going to be soluble, globular, and compact in shape, right? That's what the structure is going to be looking like. So enzymes are proteins. That's the first thing we have to remember. Second thing, they are globular proteins. They have a tertiary structure and Now, if you guys remember what we studied in the topic of biological molecules, we talked about how a primary structure is composed of a particular sequence of amino acids, right? So each enzyme is going to have a particular sequence of amino acids in its primary structure. And that is the reason that the enzyme, when it's going to undergo secondary folding and tertiary folding, when it's going to go, go and undergo tertiary structure, that specific sequence of amino acids mean that it will have specific R groups. Those specific R groups mean that it's going to have specific unique type of uh, bonding, right? Ionic bonding, hydrogen bonding, hydrophobic interaction, whatever. So those different kind of bonding are the reason that enzymes have different active sites. And if there is a mutation in the DNA, that will lead to change in the amino acid of the enzyme. And if there's a change in the amino acid of the enzyme, it will change the interactions that we are seeing in the tertiary structure, and that will change the shape of the enzyme. So that is really important, right? If we change the shape of the enzyme, then the enzyme will not be functioning anymore. And later on, you will see when we talk about denaturation of enzyme, so how does that happen as well? So an enzyme, because of this particular arrangement of amino acids, because of this one particular way that the amino acids are arranged, enzymes have this particular kind of bonding, right? They have this particular kind of active site. So the shape of the active site is going to be specific depending upon the type of enzyme it is. And now we know the reason why. Because of the primary structure, the sequences of the amino acids that are present. The sequence will dictate the secondary as well as the tertiary structure. And the tertiary structure will dictate the shape. So the shape is specific to the enzyme. Now, when we talk about the lock and key model, in the lock and key model, we studied that the active shape has one particular shape. That's it, right? The enzyme has an active site that has a one particular fixed shape. The substrate has to be exactly complementary to it, or the substrate has to be exactly matching that um, active site, right? And only when the substrate exactly matches the active site of the enzyme, when the substrate is exactly complementary to the active site, what is going to happen? The substrate will bind to the enzyme. You will get an enzyme substrate complex, and that enzyme substrate complex will then turn your substrate into a product. That's what we have studied. That was our lock and key model. That was the older theory that we believed in. Now, the newest theory that we're dealing with is actually the induced fit model. The induced fit model actually states that the shape of the active site is not fixed. So the shape of the active site is still specific. I'm not saying that it's not specific. It's still specific. It is still close to what uh what the shape of the substrate, but it's not 100% a match. What actually happens is that so the shape of the active site is close to the substrate, but it's not 100%. It's not fixed. When the substrate attaches to the enzyme, the shape of the active site modifies slightly so that the substrate and the enzyme match 100%. And then you will get the enzyme substrate complex, and then the product is made, right? That's how the reaction takes place. That's how in the induced fit model, that's how it actually works. That's the newest theory and a more accurate theory that we now understand because we've studied enzymes in more. So after studying enzymes in more detail, we now know that the shape of the active site is not 100%, not completely complementary to the shape of the substrate because that's what the locking theory was. That the active site is a completely complementary substrate. That's not true. The active site does have a specific shape and it does match or it is complementary to the shape of the substrate, but not to 100%. It is close to it. So when the substrate will fit into the active site actually undergoes modification. And when it undergoes modification, so then it becomes completely complementary to the substrate. So the site will become when a substrate will bind to the site, that's the site will modify shape. This change in shape, so it completely complements the substrate, and the enzyme reaction will be catalyzed. Now, this induced fit model, you guys will see that we can use this model, to explain a topic that we're going to cover later on, right? I mean, by later on, I mean later on in this chapter. In this chapter, we're going to study about something known as inhibitors. Inhibitors are those enzymes that those substances that can inhibit or stop the action of an enzyme in various different ways. And one of the ways that we're going to study is that substance can bind with the enzyme, and after binding with the enzyme, it can occupy the active site. So once the active site is occupied, of course, the substrate will not be able to bind. That's one of the ways. So now we can actually see that it's not necessarily that only one substrate will be binding to one particular enzyme. If you've got two different substrates, but the shapes are close enough, shape of the substrate are close enough to the active site, both of them will be able to bind to the enzyme, even if they're completely different from each other. The log and key model suggests that for one enzyme, there can only be one substrate. That's it. With the induced fit model, it has to be a closed match. It doesn't have to be a full match. It has to be a closed match. Even if you have two different substrates that are very, very close in shape, we will be able to bind to the same enzyme. We both will be able to bind to the same enzyme. Shape of the active site, since it is a close match to both the substrates, is going to be able to modify its shape in such a way that it can go ahead and it can bind to both. So, and we will go over the topic of inhibition, I will explain all of that in more detail. But that is what your induced fit model is. So that is the two theories that we know, and the induced fit model is a newer, more accurate theory. Now, we need to talk about how an enzyme basically works, right? How what is an enzyme doing? What is a specific feature of an enzyme that allows it to do what it is doing? The way enzyme works is, of course, it is catalyzing the reaction. That's fine, but one of the major things it does is that it lowers the required activation energy, right? It lowers the required activation energy that is needed for a reaction to happen. And by lowering the required activation energy, it provides basically, it lowers that required activation energy. As a result, the substrate can be converted into product. Now, when we talk about our enzymes can be both anabolic and catabolic. Anabolic means that we can use them to build up a product. So if I can use those enzymes to join two molecules together to give me, for example, a polymer. Right? I can also have catalytic enzymes. Catalytic enzymes are those enzymes that are going to go ahead and break down a polymer or break down a large molecule into a smaller. So example of catalytic enzymes, you guys have already studied your hydrolytic enzymes, your peptidases, your amylases, maltoses, all of these lipases, all of these are catalytic enzymes. But enzymes necessarily do not just break down substrates, they can also join two substrates together to give a larger molecule. So that is also possible. You can also have anabolic enzymes. Any confusion about what we're discussing so far? Is there anything that anybody would like to discuss about what we've talked
No.
about so far? Okay, all right. Now, the next thing that we have to go ahead and go over, hold on, just give me 1 second. Just give me 1 second, hold on. when the next thing that we have to basically go ahead, next thing we have to talk about is now. I mentioned to you guys that the enzyme activity can be affected by different things. The activity of the enzyme can be affected by different factors. And we have studied those factors before as well in your IGCSE, you have already done that. But now, since we have a very good knowledge of our structure of protein, the structure of enzyme, we are going to be able to understand those factors much better. Now, factor number one, what affects the activity of an enzyme, right? One of the common factors that affects the activity of an enzyme that you and I know about is that, of course, we have temperature. Now, if you talk about temperature, if you draw a graph for temperature, you would, for example, go ahead and see something like this. You would see, you would see that the temperature and the, sorry, I wrote it the other way around. Over here, I have my temperature, and over here, I have my force of reaction, or I have enzyme action. Now, you would see something quite interesting. You would see that the graph is going to look something like this. Now, uh, this you guys already know from your IGCSE that the temperature affects your enzyme activity in a very good way, right? As you're going to increase the temperature, the activity of the enzyme increases. And there's a reason for that. The reason the activity of the enzyme increases is because as I as I increase the temperature, the kinetic energy of the enzyme of the substrate increases. Since there's going to be greater kinetic energy of the enzyme substrate, they are going to be more collisions between the enzyme and the substrate, right? The more likely the substrate is to collide with the enzyme and to fit into the active site. And as a result, you will see that the rate of reaction is increasing. The enzyme activity is increasing. Now, increase will continue to happen until we reach a point that you and I had previously discussed as our optimum temperature, right? That's what we have discussed. We have a point, and this point is going to be known as our optimum temperature. Now, when we talk about this optimum temperature situation, we are going to see something quite interesting. We are going to see that you're going to reach an optimum temperature. After we stop optimum temperature, as you know, what happens? The enzyme starts to degrade. Now that we have information, now that we know about the protein structure, we know about the structure, we can understand why which pathway it has. Why is it that your enzymes are going to get deleted, right? We can go ahead and so when we talk about it, what basically happens is this, that when we increase the temperature, in our tertiary structure, we studied quite a few bonds, right? We studied ionic bonds, we studied hydrogen bonds, we studied we studied ionic bonds, hydrogen bonds. We have disulfide bridges, right? Which are strong, which are between the cysteine residues, cysteine amino acids. Between them, we have our disulfide bridges, we can have hydrophobic attraction. Now, what happens is that in the increase of temperature, these bonds in particular are, right? So disulfide bridges can be broken by the so your ionic bonds or hydrogen bonds can be such as temperature. So what happens is that when I increase the temperature, these bonds are going to be disrupted. And the disruption in these bonds actually ends up leading to the state of the active site itself being changed. Because the entire reason the active site had the shape that it had was because of all these bonds, right? and these bonds are, hold on guys, just one. So when we talk about the connection, these bonds are going to be disrupted. And the disruption of these bonds specifically leads to the change in the active site or both sides. And since now active site has been changed, the bonds have been disrupted, the surface structure is no longer what it used to be, the enzymes are simply not going to be able to bind to it. And since the substrate is no longer fitting into the enzyme, even if we talk about an induced fit model, the disruption is so severe either the enzymes are no longer sensitive or the substrate is no longer fitting into the enzyme. The consequence is that ultimately enzyme is now considered denatured. That's what the denaturation of enzyme is actually all about, right? That's how it actually works. Now, our optimum temperature, of course, in our body is around 37 degrees, right? If you have organisms that have different optimum resting body temperature, they will have different optimum temperature. In our body, our optimum temperature is going to be around 37 degrees centigrade. That's when our enzymes are going to be able to function the best. It is not only because one, they will have enough kinetic energy where the collisions between the enzymes are going to be successful collisions. I mean, majority of the enzymes are going to have successful collisions, and maximum number of enzymes are going to be involved um, in the process of reaction, right? Majority of the enzymes, nearly all of the enzymes are going to be busy or they're going to be occupied, they're going to be involved in doing the process of the reaction. So that is your temperature. Now, pH will give you a somewhat similar graph, right? If you're going to go ahead and take a look at pH, it's going to give you a close to similar graph. You're going to see over here, I have my pH. Hold on. Over here, I have my pH. And over here, I have my fourth enzyme active. So again, you're going to go ahead and observe that as we are going to again have an optimum. And I will explain the logic behind the pH as well. But before I do so, let me point something out. The optimum temperature is kind of fixed, right? The optimum temperature is kind of fixed. For example, in our body, the optimum temperature is 37 degrees centigrade. That's the temperature at which they're going to be working their best, right? But when we talk about your scenario, or when we talk about that particular scenario where we have pH involved, now when we have pH involved, the optimum pH will depend upon the type of enzyme. In which part of the body the enzyme is doing its job, right? In which part of the body does the enzyme have to work? So, for example, if the enzyme has to work, um, if your enzyme has to work in the stomach, so it needs to be able to function at an acidic pH. Similarly, if you have an enzyme that's working in the intestine, it needs to be able to work at a more alkaline pH. So the pH is going to be specific for enzyme to enzyme. So the optimum pH is not a fixed value. You can't take one fixed value and say, this is the value for optimum pH. This is the value at which we're going to be working, right? That's not how it works. We're actually going to have different values for different kinds of enzymes depending upon where they need to do their job. So when we talk about our this entire concept of enzyme, right? So again, we are going to see that as the pH is going to change, if the pH is too low or if the pH is again the why behind it, right? We can understand why is it that the enzyme is getting denatured. The reason your enzyme is getting specifically denatured is because your enzyme, is the reason for that is your enzyme, specifically speaking, has ionic bonds, right? It has hydrophobic interaction. It has ionic bonds. It has hydrogen bonds. Specifically, ionic bonds, especially, are affected. Ionic bonds are the ones that get affected, especially when pH is low. Because what is pH? pH is all about how acidic or alkaline it is, right? And that all depends upon hydrogen ion. So what actually happens is this, that the hydrogen ions, when you go ahead and talk about it, those hydrogen ions concentration changes. pH is going to be too low, we will have too many hydrogen ions, it's too acidic. pH is too high, it's alkaline. We don't have enough hydrogen ions. That causes disruption in the ionic bond. And again, now we can understand, since there's disruption in the ionic bonding, what is going to happen? You're basically going to end up seeing a particular situation where that disruption of ionic bonding is going to change the tertiary structure, and it's going to change the tertiary structure, which is going to then affect the shape of the active site, and then that is going to go ahead and prevent the enzyme from binding to the substrate, and hence the enzyme activity will reduce. So at the optimum pH, the concentration of hydrogen ions is 50%. Hence, the ionic bond are exactly as they should be. We go left or we go right of the optimum pH, we change that hydrogen ion concentration, we disrupt the ionic bond, we disrupt the tertiary structure, we disrupt the active site, enzyme activity will reduce. And again, the optimum pH is going to be different for different enzymes because it's going to depend upon where they are working. What kind of area are they working in? Are they working in a more acidic area or are they working in a more alkaline area? Depending upon that, they're going to have different optimum pH. Making sense so far? Any confusion about what we've discussed?
No.
Aruj, I hope everything is all clear so far. Like there's no confusion about what
Yes,
we
miss.
talked about. All right. Now, when we talk about it, okay, hold on. Give me a second. Let me just share this with you guys. 1 minute. So, basically, when we go ahead and we talk about our enzyme, right? We have to go ahead and so okay. Another factor, by the way, that affects enzyme activity is enzyme, right? You're going to go ahead and that greater the concentration of the enzyme, faster your reaction. The reason for that is because you have availability of your active sites are free and your active sites are available. Since the active sites is free and available, there's more enzyme, bigger concentration of enzyme, more enzyme available. What's going to happen? That as a result, the enzyme active high or rate of reaction is going to the high. Similarly, we can talk about substrate. Now, if I talk about substrate function, I will actually a graph like this. I will see that my as my substrate initially I will see that as my substrate concentration increases, the rate of reaction increases, right? And initially you will see that the rate of reaction, increase in rate of reaction is high, right? The initial rate of reaction is quite high. How is it quite high? It's quite high because I have a lot of substrate available. I have a lot of substrate that is free. I have a lot of substrate that is available for the reaction to take. Now, as I increase the substrate concentration, the rate of reaction continues to decrease, right? But if we take a look at the gradient of the graph, Initially, you will see that the gradient of the graph is a bit high. We have a bit of a steeper graph. But as the reaction continues to happen, as the reaction continues to take place, so as I increase the substrate concentration, the gradient of the graph becomes less steep. It becomes a bit horizontal. And eventually, I end up reaching a time where increasing the substrate concentration will no longer increase the rate of reaction. Why is this happening? The way it happens is this, that at the start of the reaction, my enzymes completely free, all the active sites free, all the active sites are available for the reaction to take place. But as I increase my substrate concentration, those active sites are slowly going to start to become oxidized, right? The number of free active sites will start to decrease. So the gradient of the graph is decreased. Eventually, you will reach a point where you are going to see that all of the active sites are now occupied. Once all of the active sites are full, it doesn't matter how much substrate I add. I will not be able to increase the speed of reaction anymore, right? Because my enzyme is limited. My enzyme is a certain amount. That's it. That's all the enzyme that I have. And as long as my enzyme are of specific quantity, they can only work at a specific value. Only when the enzyme has done its job and the active site is free, the next enzyme will be able to, next substrate will be able to adapt, right? And then again, only when the substrate has turned into product and the enzyme is free, the next substrate will be able to adapt. So the enzyme or the active sites are limited. So when you reach that state where you're going to see that increasing that we increase the substrate concentration, the rate of reaction does not increase anymore. You'll reach a plateau, you'll reach a flat horizontal area. Increasing the substrate concentration no longer increases the rate of reaction. This is a point at which all the sites are occupied, all the active sites are fully occupied, enzymes are working at their maximum possible speed. That point is known as Vmax. So Vmax is the point at which the enzymes have reached their maximum activity because all the enzymes are basically occupied, all the active sites are basically occupied. Now, when you're going to go, so these are basically the factors that we have to go ahead and talk about. So we have to know about pH, we have to know about enzyme activity, we have to know about our temperature, we have to know about substrate concentration and our enzyme. Now, let's discuss about something that is definitely new for you guys. What is new for you guys? And where would we need an inhibitor? So an inhibitor can be something that we want to happen or something that we don't want to happen. For example, an inhibitor, when I want it to happen, let's say there's certain enzyme that is I need that is causing a disease. For example, in our nervous system, what happens is that we can have certain kinds of diseases in which the nervous system is not working properly, right? And we'll talk about that later. In those diseases, what happens is that I have enzymes that break down the neurotransmitter. Neurotransmitter is a chemical compound that allows the nerve impulses to conduct from one nerve to another. So, in certain situations, in certain diseases, I want to make it so that I want the neurotransmitter to be working for a longer time. So, as a result, I can give inhibition. Then that inhibitor will act on the enzyme. So the enzyme does not break down the neurotransmitter. So our nerve impulses can be conducted in these situations. So that is an example of where I want to use an inhibitor, where I want to work with an inhibitor. We can have situations where inhibitors can also be something we don't want. And I'll give you an example. An example of this is you guys must have heard that you do not take medicine for your own, right? There's a reason for that. The reason for that is often you must have heard people say drugs can interact with each other. What does that mean? I mean, what is that? How do drugs interact with each other? They work like this. So, of course, when you eat any medicine, when you eat a drug, when you're taking a drug, the enzymes are responsible for breaking it down. Now, if two drugs are similar in shape, both of them can go for the same enzyme. And since both of them can go for the same enzyme, what can happen? The breakdown, one of them is, of course, let's say one of them has stronger attachment or stronger attraction for the enzyme. So that drug will bind to the enzyme. The other drug will not bind. What will happen? The drug that will not bind will stay in the body for a longer period of time and it will produce side effects. So that is an example of an inhibition that we don't want. And then we have inhibition that we do want, where we actually use inhibitors for medical treatment. Now, what are the two types of inhibitors that you need to know? We have competitive inhibitors and we have non-competitive inhibitors. What is the difference between the two of them, right? First of all, I think you guys can just take a look at the word and figure out what the difference between the two of them is. But actually speaking, it goes slightly more deeper than that. In competitive inhibitor, what happens is that I have two substrates that are similar in shape. This is what I was talking about when I also mentioned this in induced fit model. So in induced fit model, I also mentioned it to you that the shape of the active site is modifiable, right? So what happens is that you have got two substrates that are able to fit into that particular active site because they are similar in shape. So they are both complementary to the shape of the active site. So what will happen? They will compete. They will compete with each other for the active site of the enzyme. And that's the logic I can use in my medical treatment. If I'm giving a competitive inhibitor, so what can that competitive inhibitor do? It will compete. The substrate, for example, I gave you guys the example of neurotransmitter, that I don't want the neurotransmitter to be broken down. I don't want the enzyme to act on the neurotransmitter. So I can go ahead and give them, I can give this competitive inhibitor. The competitive inhibitor will be competing for the active site. It will bind. So instead of letting the enzyme bind to the neurotransmitter, the medicine or my drug will bind to the enzyme. So now the active site of the enzyme is occupied by my drug. So the active site is no longer free to bind to the neurotransmitter. So that will inhibit the activity of the enzyme. So it's not inhibition in the sense that the enzyme stops working. It's inhibition in the sense that the enzyme is not going to be working on that particular substance, right? This is a competitive inhibition. When a competitive inhibition, the active site is involved, right? The active site is going now. The other thing about competitive inhibition that because again, right, I say that I think there is more neurotransmitter competitive inhibition. They're both in the same in terms of your in both same in terms of your space. Both have the ability to bind both of them. But since the competitive inhibitor, since the competitive inhibitor is going to be since the competitive inhibitor is now lesser in quantity, The other substrate is now higher in quantity, so the substrate will be able to bind to the enzyme once again. This sort of inhibition is a reversible, right? This inhibition is a reversible inhibition because it depends upon quantity. If the inhibitor is in more quantity, then yes, we can go ahead and we can definitely uh, the inhibitor will be binding to the active site because inhibitor is higher in concentration. So between the competition, the inhibitor wins. But if the substrate increases in concentration, again, what's going to happen? That the substrate is going to win, right? So this is a reversible process. It's not a permanent type of inhibition. If you want to reverse the inhibition, simply increase the substrate. That's all you have to do. Let me give you guys another example. I give you guys the example of neurotransmitter. Let me give you guys another example. Another example that we have is that let's say that okay, there is something that is known as NPC. It's used quite commonly, in your refrigerators and all that to basically make sure that it doesn't freeze over. It's something that's used quite commonly. You guys might have heard of it or might not have heard of it. We have antifreeze. Antifreeze is basically chemically it's ethylene glycol. That's what chemically it is. Now, what if a person accidentally drinks antifreeze? You see this happening in kids, right? Because children sometimes accidentally end up drinking household bleaches or household chemicals that are lying about detergent. What if a person ends up drinking ethylene glycol? The treatment that we can go over is that now the problem with ethylene glycol is that the body rapidly breaks it down or the body rapidly converts it into an acid that is known as oxalic acid. Okay. Now, this oxalic acid is bad for us. It can cause severe, irreversible kidney damage. Now, what would you and I do for the treatment? Now, the enzyme, that particular enzyme that binds to your ethylene glycol and converts it into oxalic acid, it has a competitive inhibitor. The competitive inhibitor is ethanol. So the shape of ethanol is similar to ethylene glycol. So if we can use ethanol as a treatment, right? The ethanol will then bind to the enzyme. It will bind to the active site, and by binding to the active site, it can inhibit the activity of the enzyme. So now that enzyme is the active site of the enzyme is occupied by ethanol. So it's no longer free. The active sites are no longer available or the active sites are occupied by the ethanol. They are no longer available to bind to your ethylene glycol. So ethylene glycol will not be converted into the toxic product and the person will be safe. So that is an example of competitive inhibition where we are using it for our purposes. And competitive inhibition is again reversible. If the person got more ethylene glycol for whatever reason, the effect of ethanol would no longer work. Now, the other type of inhibition that we have is that we can have non-competitive inhibition. Now, in non-competitive inhibition, we can have again, it can be a reversible inhibition or it can be a non-reversible. Okay? In non-competitive, we can have both of them. We can have reversible inhibition and we can have non-reversible inhibition. Both things are possible. Let's go ahead and take a look. Let's go ahead and talk about. When we talk about our non-competitive inhibitors actually, so you're going to go ahead and see that. You're going to go ahead and see that in your non-competitive inhibitors, you are actually going to, okay, so in non-competitive inhibitors, we have multiple options. Okay? Multiple things can happen in non-competitive inhibitors. What are the multiple things that can happen? Now, the first thing that can happen non-competitive inhibitors. Now, in non-competitive inhibition, the inhibitor will bind to some other area of the enzyme other than active site. So in competitive inhibitor, they were all competing or they were all fighting for the same active site. In non-competitive inhibition, that doesn't need to happen. In non-competitive inhibition, that doesn't need to happen, right? In non-competitive, basically because in non-competitive inhibition, uh, the inhibitor is going to be bind to some other region of the enzyme, not the active site, right? Not the active site. It's going to be bind to any other region of the enzyme. And what's going to happen, this particular area, that particular area of the enzyme to which the inhibitor binds, we call it the allosteric site. That particular site to which a non-competitive inhibitor is going to bind, we call it allosteric site. And it is, except for active site, it's any other part of the enzyme. Now, what's going to happen? How is this going to work? So we can understand how a competitive inhibitor is working, right? It's competing for the active site. It's occupying the active site. The active site is not free to bind to the substrate. What about a non-competitive inhibitor? In a non-competitive inhibitor, what happens is that the inhibitor will bind to the enzyme at the allosteric site. And after it will bind to the enzyme, it will disrupt the hydrogen bonds and your hydrophobic interactions that are hydrogen bonds, the hydrophobic interactions that are holding the enzyme in its three-dimensional globular shape, right? So as a result, what's going to happen? The distortion will ripple through, like it literally ripples, spreads through the enzyme all the way to the outside. As a result, what happens? This disruption of bond is going to disruption of bond will happen at the active site because of the ripple, because of the fact that this sort of disruption, this distortion is spreading through the enzyme. It will happen to the active site, and now your active site is um, no longer able to, you know, the active site is no longer complementary to the substrate. So the substrate will not be able to bind to the active site, and this is an example of non-competitive inhibition. No longer able to bind, no longer complement to the substrate. And this is an example of non-competitive inhibition. If I increase the concentration of the substrate, it will have no effect. In competitive inhibition, if I increase the concentration of the substrate, I told you guys we can reverse, we can stop the effect of inhibitor. That's not true for non-competitive inhibition. In non-competitive inhibition, as long as the inhibitor is attached, as long as it is attached to the enzyme, the inhibition will continue. Once it deattaches, once it leaves the enzyme, then yes, it will be reversed. Then it will reverse and it will go back to normal. This is an example of non-competitive inhibition, right? So, and actually, you will see another, when you go ahead and talk about it, you will see that inhibition of enzyme is not something that we do, or it's not something that you and I do all the time. you will see that inhibition of enzyme is something that happens naturally in our body as well. How does it happen naturally in our body? In our body, it happens naturally as well in the form of end product inhibition. How does end product inhibition work? So let's say that I have my substrate. This substrate is being converted into product one. It's a series of reactions. Then that product gets converted into another product, product two. Then that product two gets converted into the product three, that is our end product or the final product or what we were actually trying to make. So in our body, we don't just go from substrate to end product, right? For example, if I want to take glycogen and I want to turn it into glucose, I first have to use amylase and break it down into maltose. And then I have to use maltase and break it down into glucose, right? So it's not a direct process. I have to do a multiple step process. So what's going to happen that in this particular situation, this end product is going to be the one that is going to inhibit the effect of the enzyme. This end product is going to be the one that is going to inhibit the enzyme. Hold on. So let's say I have over here, I am dealing with my enzyme one, and then over here I'm dealing with another enzyme that is enzyme two. Then of course I have enzyme three. So these are the different enzymes that are involved. And I give you guys a good example. Right now, I told you guys that, for example, we have uh, starch. So when we have starch, what's going to happen with the starch? That the starch is going to be broken down into your, first it's going to be broken down into your maltose or your starch is going to be broken down into maltose. And that starch, after it's broken down into maltose, then it is going to go ahead and actually convert your maltose is going to then turn into glucose enzyme molecule. So we have multiple enzymes. Now, what is going to happen is what is going to go ahead and update. You're going to see that over there my end product. My end product is the one that's going to go ahead and inhibit the activity of enzyme. That my end product or my final product is one that is somehow or the other inhibiting or stopping the activity of enzyme. Now, why are we saying something like this? Because I told you guys something happens normally, something that happens psychologically in our physiologically in our body, right? It's a normal thing. Why is this happening? The reason that this is happening is this is because we want to have a feedback mechanism. We want to control the process, right? So what is going to happen that in the end, let's say that the end product has been made. We don't want the concentration of the end product to increase to a huge value. I don't want the end product to increase to a huge value. So this end product by itself will inhibit enzyme number one. So no more end product is made. And once all the end product is used up, the inhibition will stop, right? Because like I told you, as long as the inhibitor is attached, the inhibition will happen. Once the end product has done its job, once it's all used up by the body, for example, the inhibition will stop because now there's no more inhibitor. As a result, the reaction will start all over. So this is end product inhibition, where the end product itself is inhibiting the enzyme. And why is it doing that? It's doing that because we want to make sure that we don't end up getting too much end product. For example, I don't want to end up too much neurotransmitter in a healthy person because that might damage the system, right? So that is end product inhibition, where the actual final product of the reaction itself inhibits the enzyme. And why do we want to do that? To regulate the amount of end product we are getting. So once the end product is used up, the inhibition stops and the process will start again, and you will be able to make the end product once again. Making sense what we discussed so far? Any confusion what we've talked about?
No.
Arush, I hope everything is clear. Okay, guys, if everything is clear and nobody has any questions, we'll go ahead and stop it over here for today. We are just going to go ahead and start in the next class, finish off our topic. Next class, what we have to do is something that is a bit newer and a bit tricky. That's the only tricky thing in this particular chapter that we have to go over our Michaelis Menten constant. So, inshallah, we'll go ahead and take a look at that in the next class. All right, guys?
Okay, miss.
Okay, thank you so much, guys. I'll see you in the next class. Have a good day.
Bye, miss.
Bye, guys.